Welcome to Lecture 21, where we will go over neurons and their role in the nervous system. A neuron is a cell that acts to receive information at the location of the dendrites that are located near the cell body. Neurons will then transmit this information along the axon, and eventually it will reach other cells. At the location where it reaches other cells, whether these be neurons or other target organs such as muscle cells or liver cells, where these cells meet, we have the synapse. There are three main stages in information processing. These involve sensory input, integration of the information, and motor output. Sensory input involves the sensory neurons that detect information from the surrounding environment. These will then relay this information through the use of interneurons, either to the brain or to ganglia that are distributed throughout the body. The brain and ganglia are responsible for processing this information and relaying any of the information to the effector organ to initiate a response. The motor output stage of information processing involves when the motor neurons are activated. These will then be stimulated to cause a response in the animal. There are three main types of neurons. The structure of a neuron is diversified with respect to its overall function within the body. For instance, sensory neurons typically have many dendrites and a very long axon. Interneurons, on the other hand, have highly branched dendrites. As a result, they can receive input from many different synapses. And finally, motor neurons have highly branched axons. This will then allow them to transmit information to many different target cells at the same time. Nerves consist of groups of axons from many neurons that are arranged in bundles. Many animals have their neurons arranged into two main types of systems, the central nervous system and the peripheral nervous system. The central nervous system, or CNS, has neurons that can sort, process, and integrate information. These are typically arranged into a cluster of ganglia found throughout the body, or arranged into a brain, which is typically located at the anterior end of the body. The peripheral nervous system, or PNS, have neurons that carry information from the CNS to the rest of the body. Both the CNS and PNS have what is termed glial cells. These glial cells are important in supporting the surrounding neurons. Note in the image to the right, the glial cells are present in red. These support the interneurons that are present within this rat brain. We are now going to examine how a neuron works. Around a neuron, ions are unequally distributed across the membrane. Overall, there is typically a negative charge inside a neuron. The differences in charge across a membrane leads to a membrane potential. The resultant attraction of opposite charges across the membrane is also seen as a source of potential energy. The membrane potential and the voltage across that membrane typically involves the movement of sodium, potassium, and chloride ions. The resting potential refers to an inactive neuron that is not sending a signal. Note that the typical resting potential of an inactive neuron is negative and sits between negative 60 to negative 80 millivolts. Changes in the voltage across the membrane will result in what is termed an action potential. This action potential will then be passed along the axon. This is how neurons carry information throughout the body. The resting potential of an inactive neuron sits between negative 60 to negative 80 millivolts. This is established mainly through the action of two different kinds of membrane pumps, ion channels and the sodium-potassium pump. The sodium-potassium pump is important for maintaining a high concentration of potassium inside the cell. If you recall, the sodium-potassium pump will actively transport 3 sodium ions out of the cell for every 2 potassium ions brought into the cell. This will help to establish a high concentration of potassium inside the cell that the ion channels can then take advantage of. Ion channels are also present that help to establish the net negative charge inside the cell. This occurs as many potassium ions will flow quickly out of the cell due to the gradient that was established from the sodium-potassium pump. If you take a look to the diagram to the right, note the number of potassium channels far exceeds the number of sodium channels. The ion channels help to convert the chemical potential energy from the gradient that's established into a voltage or electrical potential energy. We're now going to take a look at the type of response that is generated within a neuron. These can be termed as graded potentials or an action potential, which we will take a look at in a few slides. In the graded potential response, when a stimulus is exerted on a neuron, the membrane potential of the neuron can change, mainly due to the action of gated ion channels. The strength of the stimulus can often be reflected in the magnitude of the shift in the membrane potential. Graded potentials can either lead to hyperpolarization or depolarization. In hyperpolarization, the membrane potential gets more negative. For instance, it goes from a resting potential of minus 80 to minus 90 millivolts. In a hyperpolarization event, stimulus causes the voltage-gated potassium channels to open. This causes even more potassium to leave the cell. In a depolarization event, the membrane potential becomes less negative. This is because a hyperpolarization event will lead to the opening of the sodium-gated ion channels. As a result, the inside of the membrane will become less negative as sodium will enter into the neuron. Graded potentials in general dissipate as they move down the axon. This means that they can decay over time and also as distance increases. So graded potentials are only good for a short distance and a short time. This diagram represents the different types of graded potentials: hyperpolarization and depolarization. Note in the hyperpolarization, we have the inside of the membrane getting more negative, whereas in depolarization, we get the resting potential becoming less negative. Also note that there are two different types of responses that vary with respect to the magnitude of the membrane potential change. Hyperpolarizations, remember, are due to an increased permeability to potassium, while the depolarization events are due to an increase in permeability to sodium. We're now going to take a look at the action potential response of a neuron. If the depolarization event reaches a threshold level of about negative 55 millivolts, an action potential within the neuron can be generated. Note that this is considered an all or none response. This means that if an action potential is generated, the magnitude of that action potential is not correlated to the strength of the stimulus, as we saw in the graded responses. As the minimum threshold of negative 55 millivolts is reached, a positive feedback will trigger the opening of more voltage-gated sodium channels. This will cause further depolarization, while the potassium channels will close. As the peak voltage for the action potential is reached, sodium channels become inactivated, and the potassium channels open. This means that the inside of the membrane becomes negative. There is an eventual lag while the sodium channels close, followed by the potassium channels, causing an undershoot in the action potential and in the voltage charge. As the potassium channels close, the resting membrane potential is eventually restored. We're now going to take a look at the different phases of the action potential. We begin with the resting state. Note that in the resting state, the gated channels for sodium and potassium are considered closed. Note that the ungated channels, like the ion channels for sodium-potassium and the sodium-potassium pump, are all open to help maintain the resting state. When a stimulus is encountered, this will open some of the sodium channels. This will cause sodium to flow into the cell and depolarizes the membrane, causing it to become less negative. If this depolarization reaches the threshold of negative 55 millivolts, this will trigger the action potential. This would lead to the rising phase of the action potential. During the rising phase, depolarization opens most of the sodium channels, which allows sodium to rush into the cell. However, the potassium channels typically remain closed. This leads to a rapid depolarization event. After we reach the peak of the action potential, we enter into the falling phase. During the falling phase, most of the sodium channels become inactivated. This will block the sodium influx into the cell. Most potassium channels remain open, permitting potassium to flow out of the cell. This will help to make the inside of the cell negative again. However, there is an undershoot. As the sodium channels close, some of the potassium channels still remain open for a small period of time. As these potassium channels close and the sodium channels become unblocked, the membrane will then return to its resting state. Within the firing of an action potential, a refractory period occurs. This refractory period occurs when the sodium channels are inactivated. This creates downtime within the neuron when a second action potential cannot be generated. When transmitting information, a series of action potentials will be carried down the axon. This will allow for the original nerve impulse to travel from the cell body to the synaptic terminal to cause a response. Since the action potential is considered all or none, the magnitude of each potential is consistent as it travels down the neuron. Note that this would not happen in graded potentials, as the magnitude of the potential in a graded response dissipates over time and as well as over distance. The axon hillock within a neuron refers to the location where the action potential is initiated. At this location, the sodium will flow inward, creating the rising phase of the action potential that will depolarize neighboring regions on the axon. This will reinitiate the action potential there. The original location where the neuron is initiated will repolarize as potassium flows outward. This depolarization and repolarization is repeated as the action potential is propagated or spreads down the axon. This slide gives you a visual representation of how an action potential spreads down the axon. At the axon hillock is where the action potential is initiated. When the action potential is initiated, sodium ions will flow inward into the neuron. This depolarization will then spread to neighboring regions. However, there will be a repolarization event at the initiation site. This depolarization-repolarization will be propagated along the axon until it reaches the synaptic terminal. There are many adaptations that occur in neurons. Natural selection in general will favor quick responses. This will allow for an organism to quickly respond to changes in their environment, such as the presence of predators, danger, or to respond to opportunities that may arise, specifically with respect to prey capture. Some of these adaptations include wider axons. Wider axons provide less resistance to the current that is generated as action potentials pass down the axon. This will allow for the action potential to travel much more quickly down the axon. Wider axons are present in giant axons that are in many invertebrate species such as squid. This allows for rapid responses and will allow the squid to propel towards a prey item very quickly. Another adaptation found in neurons is the presence of electrical insulation. Electrical insulation on the neurons will allow for action potentials to travel further. This will then have an overall increase in the speed of how the action potential is propagated down the axon. Myelin, which is present in vertebrate axons, provides insulation to help increase the speed of a traveling action potential. Myelin is a lipid-based sheet that can be deposited around a neuron. It's typically deposited by specialized support cells, such as oligodendrocytes in the CNS and Schwann cells in the PNS. Myelin is considered to be a poor conductor, thus it has an insulating effect on the neuron. Between these areas of myelination, there exists the nodes of Ranvier. The nodes of Ranvier are important locations on the neuron. This is where most of the voltage-gated sodium channels will be received. This also provides a point of contact between the neuron, the membrane, and extracellular fluid. As a result of the nodes of Ranvier in combination with the myelinated areas, the action potential will be regenerated at the nodes of Ranvier. This allows for the action potential to travel very quickly down the axon. This is because the closing of channels is limited to specific regions, and thus it is concentrated at the nodes of Ranvier. Myelinated axons can be quite thin, allowing for many myelinated neurons to be present within an area. This will also help to increase the speed of transmission. Schwann cells are present in the PNS that will help to secrete the layers of myelin. Remember, oligodendrocytes would be present within the central nervous system. Note the presence of the myelin sheath, which is deposited by the Schwann cell. The Schwann cell itself will be kept within the layers of the myelin. Also note the presence of the nodes of Ranvier. The depolarization and regeneration of the action potential is restricted to the nodes of Ranvier. This will help to increase the speed of the propagation as the action potential jumps down and travels down the axon.